thank you for braving the uncertain weather today to come and listen. I'm going to talk about plant pigments in relation to eye health. Now, as nutritional scientists, epidemiologists, and physicians, we have focused on single nutrients, often energy-yielding nutrients like fat or vitamins and minerals that are related to slowing chronic age-related diseases. And it isn't until the last decade that we've been realizing we're missing a whole lot. We're missing a lot of the bioactive phytochemicals in plants that can preserve eye health as well. And so today's discussion is about one specific plant pigment as an example. And you can see we'll start with a focus on a role for this specific plant pigment, but also put it in the context of broader diets by the end of the conversation. There are several age-related conditions that become more common as we age. The most common are age-related cataracts and macular degeneration. And we will be focusing on these plant pigments, which are lutein and zeaxanthin, and their relation to these conditions. There has been some work in relation to other conditions such as diabetic retinopathy, but this is much less studied. So we'll focus on this pigment in relation to age-related cataracts and macular degeneration. As you can see from this slide, these chronic age-related conditions share a lot of pathogenic mechanisms of many other things that creep up as we age and kill us. Oxidative stress, inflammation, hypertension, hyperglycemia or insulin resistance, in some cases angiogenesis, but they are unique. The pigments we'll be talking about are unique in that they address another pathogenic mechanism that's unique to the eye and actually a little bit different from the photo damage caused by the skin. This is photo damage. So light gets in our eyes so that we can see, but over age, as we live longer, it has deleterious effects which need to be protected against. I want to start with a broad background slide that tells you that these are not the only things that we've been studying in relation to age-related eye disease. And if you look at this timeline, you can notice that what we have studied in relation to eye disease closely mirrors what other people have studied. I'm still getting the hang of this. There we go. What other people have studied in relation to heart disease and cancer and many other chronic conditions. So epidemiology of age-related eye diseases really took off in the late 1980s, largely because of the results much on this campus, the ability to take photographs of the lens and the retina and grade them systematically and study them in relation to risk factors. And so we and others have studied various antioxidants in relation to these conditions, and that was what was being studied with regard to cancer and heart disease as well. Also, there was a period in the 90s that specifically focused on dietary fats as risk factors, refined sugars. There was an area where we were focusing on folate. We and others have studied that and B vitamins in relation to these diseases. And more recently, there's been a focus on anti-inflammatory nutrients and putting the whole package together, realizing that there are many of these nutrients and diet components that are related and all contribute towards possible reduction in risk for many diseases. So we'll be focusing on lutein and zeaxanthin, two plant pigments where a lot of our research has focused in the last decade. We have also done studies with regard to vitamin D, which I will only briefly mention, but I do hope to also bring in some discussion of our work on overall healthy diet patterns in relation to these age-related conditions. So we'll talk first about cataract, then macular degeneration, and as time permits, some of the future directions that we're interested in. Okay, they're carotenoids. They're one of the three major classes of plant pigments. There are hundreds of them in nature, over 500. And they're responsible for many of the colors we see in nature, for many of the smells in nature. Of the hundreds, only 50 of them are in the foods we eat. And of those, Only these 5 in significant quantities are accumulated in human blood. Now, we've been focusing on the carotenoids, which can be converted to vitamin A in the last couple of decades, such as beta-carotene, alpha-carotene, and cryptoxanthin. And lutein and zeaxanthin is very chemically similar to these, almost identical except for a hydroxyl group. But you can see that despite the similar chemistry, they're very different in color and very different in the way they're accumulated in both animal and plant tissues. So lutein and zeaxanthin are yellow pigments, different than the orange pigments that the carotenes impart or lycopene imparts. They accumulate, they're responsible for the yellow color of corn. And they also accumulate in eggs, so they're largely responsible for the yellow color that are in eggs. But they're in highest concentrations in dark leafy greens. We don't see them because dark leafy greens also have chlorophyll. They tend to accumulate in areas that are sun exposed, just as chlorophyll does. But if you boil spinach or you boil kale, you see that the water's yellow. And those are the pigments lutein and the structural isomer zeaxanthin. These carotenoids represent 25% of the pigments which are in diet, in blood, and most tissues of the body. But, and also in these tissues, there's more lutein than the structural isomer zeaxanthin. But in the eye, they're the most abundant carotenoids throughout the eye. And also the concentration of zeaxanthin is much higher. They accumulate especially in the area of the retina called the macula, which is the region that macular degeneration develops, where they're 1,000 to 10,000-fold higher in level than in the blood, and about 100-fold higher than in the lens and in other areas of the eye. And so we think that there might be, not in addition to a photoprotective effect, there might be some effect on vision. And I'll just get into that briefly at the end of the discussion if there's time. But let's first talk about these in relation to lens health and then macular health. Okay, cataract is the #1 cause of blindness worldwide. In the US, it's not so because we have freely available cataract surgery. But it is the leading cause of vision loss in young old, that is people about 65 to 75, because about 30% of people in this range have vision-limiting cataracts. And cataracts are simply a pacification of the lens, which results from protein denaturation over time, and it scatters light then at the back of the eye, causing us to see in a more blurry way. And also, it's first noticed when we're driving. Often it's as contributing to glare. It's very common. It really is happening in all of us. It begins happening in our 20s, slowly, in different rates and in different people. By age 75, half of us either have a visually limiting cataract or have had cataract surgery in at least one eye. So surgery accounts for a large proportion of our Medicare budget. Last time somebody estimated it accounts for 12%. It's also been estimated if we can slow the development of cataract, we can cut the need for surgery in half because more of us would not need to have cataract surgery in our lifetime. So potentially large public health importance. We develop cataracts in many regions of the lens. We're going to focus on cataracts that develop in the center or nuclear area of the lens, and I'm not physically coordinated enough to use this pointer very effectively here. So I won't use it. So we'll focus on cataracts that develop in the nuclear area of the lens, and you will see that the plant pigments I'm talking about are simply one of many risk factors for the development of cataract, and we'll have to think about it in that context. It's biologically plausible that these carotenoids would protect the lens. from damage. There have been experiments done in human lens epithelial cells that have been exposed to UV light. If they're first exposed to lutein, the oxidative damage is lower. Studies in diabetic rats have shown that if fed lutein, the progression of cataract is lower as well. And this has actually been the most consistent association of my career as a nutritional epidemiologist. Early in the mid-90s, Barb Lyle, working with us in work with the Beaver Dam that we did with the Kleins, we showed that the 5-year progression of this nuclear cataract was twofold lower among people whose diets were high in lutein and zeaxanthin. And subsequently, this result has been found in many other study populations. It's found to be quite strong in studies that use photographs to assess lens opacities, and less strong in studies that, that simply use cataract extraction as an endpoint. Probably because cataract extraction also involves health-seeking behavior, which can attenuate and bias these associations. So we have been specifically studying this association in the carotenoids and age-related eye disease study, which is this data point here. So let's talk a little bit more about this association and how it relates to other risk factors. First, a little bit about this study. This carotenoids and age-related eye disease study, or CARIDS, is an ancillary study to the Women's Health Initiative. As many of you are aware, this study was began in the mid-1990s, and there were both observational and clinical trial components, and this is in the observational study cohort. And in this cohort, thank you. And in this cohort, they had previously provided food frequency questionnaires, which were used to estimate levels of lutein and zeaxanthin in the diet and in the serum. And so we selected, kind of in reverse to a case control study, we selected exposures accumulating people with high and low lutein intakes, which were essentially people with high plant food, high vegetable diets, and low vegetable diets. And then we invited them 6 years after that for participation in our study in either Wisconsin, Iowa, or Oregon to take eye photographs to assess cataract, macular degeneration, and the density of these pigments in the back of the eye, macular pigment density. We also got more information about diet so we could understand something about diet stability as well as supplement use. So we had 3 measures of exposure to these pigments. We had measures in the diet. In nutritional epidemiology, we like to ask about people's diets for a number of reasons, one of which is we can then translate our results to levels of risk reduction that can be achieved through dietary changes. So we have estimates in the diet, but of course we know that the composition of the diet in terms of lutein and zeaxanthin and many vitamins and minerals is quite variable, and nutrient databases on which these estimates depend are sometimes inaccurate. So people often like to confirm results in diet with results in blood. With regard to these plant pigments, it has the added benefit in reflecting the various factors, many of which we don't know, that influence our ability to absorb these pigments and to transport these pigments, although the disadvantage of their shorter-term estimates than dietary estimates. But it has the disadvantage in blood levels are also affected by other homeostatic mechanisms, which may make them less useful as an indicator of status. We also then estimated levels of these carotenoids in the eye by estimating non-invasively macular pigment density, and I'll show you some of those data. So we're talking about cataract, and this shows you the age-adjusted odds ratios for nuclear cataract that were significant. And you can see that people who had diets which are low in lutein had about a 50% higher odds of nuclear cataracts 6 years later. The association was identical for serum lutein and similar but not significant when we looked at the level of lutein in macular pigment. But importantly, it's only one of many aspects of a healthy diet which might contribute to lower cataract. And in fact, intake of lutein might be a biomarker for a healthy diet in some ways. You can see that we found an even stronger association when we scored people on adherence to recommended dietary patterns, looking at people in the lowest versus highest quintile there. It was stronger. Now, it's popular in nutritional epidemiology to adjust nutrients for each other. It's not a very good idea, and there are a lot of problems with that, which we won't get into, because food is not simply a vehicle for these things. Many of them travel together. But we did that with regard to lutein, and you can see that if you take our original association with lutein and adjust it for healthy eating index score, the association is attenuated. That's not exactly kosher because part of a healthy diet is lutein. We've also looked at it the other way, adjusting healthy diet score for lutein, and it's less attenuated. So probably some of what we're observing with lutein does reflect healthy diet, and some of what we're observing with healthy diet reflects lutein. So we've talked about the evidence that lutein is related to lower odds for nuclear cataract. Now let's talk about macular degeneration. And that develops, as I mentioned, at the back of the retina in this area by the pit, also the area that has this large yellow pigment patch from the lutein and zeaxanthin that we eat. Age-related macular degeneration is fairly common in early stages. We detect it photographically in our studies like this. And you can see early signs of macular degeneration as drusen, these whitish yellowish spots, and sometimes as pigmentary abnormalities as a more progressed stage, which are darkish dots. And these can be graded for the extent of drusen. They're common, and some of us, but not all of us, will go on to develop late stages of macular degeneration involving the loss of cones, and mostly cones, some rods, and that atrophic loss is referred to as dry macular degeneration, and sometimes new blood vessel development or neovascularization, referred to sometimes as wet. When the cones are lost, it interferes with our ability to see straight out front. Our central vision is affected. And also our ability to see fine detail. This is a cone-rich area. So when we're reading the side of a pill bottle or newspapers, we're using that central area. So that's profoundly affected. There is no cure for macular degeneration, and treatment options are limited to people with the dry stages of disease. With the wet stages of the disease, which is a minority of cases. And it's costly, it involves monthly eye injections, so there's no good treatment. So this is costly to individuals and costly to our healthcare system. Again, lutein and zeaxanthin are only some of many risk factors for macular degeneration. There's low levels of lutein and zeaxanthin. The most important risk factor for macular degeneration is family history. It runs in families. That's been known for quite a while. And more recently, specific genetic variants that predict risk have been identified, particularly in the complement system, which reacts to inflammation by generating a bunch of molecules to create the inflammatory response, and then sometimes in an exaggerated way. So there are genetic risk factors that are common and predict this occurrence, and these continue to be studied. Smoking is the most consistent lifestyle risk factor associated, and these other risk factors are sometimes but not always observed. It's fairly inconsistent. Age-related macular degeneration epidemiology is inconsistent for reasons we'll touch on. So in some studies, but not others, hypertension, cardiovascular disease, and diabetes predict macular degeneration. Sunlight, there's strong biologic evidence that it should predict macular degeneration. The epidemiologic data are unclear, largely because measuring sun exposure is difficult. Some studies have found that people with higher body mass index have more macular degeneration. And this is usually, it is probably reflecting some genetic factors and some dietary factors, which are less frequently studied in relation to these other risk factors. With regard to diet, the most commonly found risk factors are diets high in fat, diets low in fish or omega-3 fats, and diets low in lutein and zeaxanthin. And I should also point out that there was a clinical trial about 10 years ago called the Age-Related Eye Disease Study, in which high-dose antioxidant supplements lowered the progression of this intermediate to advanced stages, providing evidence that oxidative stress and preventing it might be an important pathogenic mechanism. We and others have recently found that physical activity predicts lower risk for macular degeneration, and that poor vitamin D status enhances risk for macular degeneration. But I'd also like to point out that the early studies don't include many of these risk factors. There's strong biologic plausibility for lutein and zeaxanthin and protection against macular degeneration. Specifically, we know that these pigments absorb blue light, and blue light is the most common in the atmosphere. It is the light that is most damaging to the back of the eye. And there's a large body of evidence showing a mechanism for this by lowering the accumulation of toxic byproducts of vitamin A metabolism in the eye, which go on to be an inflammatory trigger. Also, in primates, it's been shown that primates who are deprived of lutein and zeaxanthin do not accumulate this in the back of their eye, and they're more susceptible to blue light damage of the fovea. We know lutein, like other carotenoids, are antioxidants, so that's a possible role as well. And there's been a lot of work showing that they have a variety of other biologic functions which we're only beginning to understand. And also, they may act not only in the eye, but systemically as an antioxidant and anti-inflammatory. And I've shown you here, this is just a picture of lutein and zeaxanthin. It doesn't look like that physiologically, but this is a picture of a macaque eye where you can see the yellow lutein and zeaxanthin pigment and how it accumulates, especially right in the center of the macula. Over the past 2 years, there's been a consistent body of evidence suggesting that people whose diets are high in lutein and zeaxanthin have lower odds for advanced macular degeneration. It can be quite strong in many studies. It's not quite as consistent for early macular degeneration. We're, of course, more interested in knowing how we can stop the progression of this condition at earlier stages, and the data have been quite inconsistent. Now, does this mean that lutein and zeaxanthin only physiologically slow progression of the advanced stages, or does it mean we're not able to observe it? Our data in the Carotenoids and Age-Related Eye Disease study indicate that there might be some biases that are causing this inconsistency, and probably a lot of inconsistency in the epidemiologic research of macular degeneration. Here's what we found. This shows the odds ratios for early macular degeneration in people whose diets are high compared to low in lutein and zeaxanthin. And you can see we found no association originally. Now, partly that could be due to the fact that we're only measuring diet at one time point, because when we looked at people who had wildly unstable diets, we saw that there was a change in the odds ratio in the protective direction. But also, what we observed is a very significant age interaction. We'd actually observed that in a couple of other samples before that. And so that if you looked at the 3/4 of the sample that was under 75 years of age, we did see a protective association, and quite the opposite for people over 75 years of age. So what do you think that indicated, students of epidemiology? Competing causes. What else? What happens as we get older? What happens if we don't eat a lot of fruits and vegetables? We're more likely to live or die. Okay. Yeah. Also, what happens if we've been told that we have diabetes or have heart disease? We eat better. Yeah, and also other risk factors. People who have hypertension die. People who smoke die. So the people that are living to be counted, the cohorts that remain, are quite different in a large number of ways. And in fact, those people often comprise the largest segment of study populations. So biases in that age group might be particularly influential, as we found they were in our sample. And we found that the reversal in association is not limited to lutein. Here I show the direction of associations of AMD differ in the younger versus older age groups when you look at quintiles of overall healthy eating index. And you can see we've divided the sample into 12 age quintiles. And we consistently see that having higher scores on healthy diets are associated with lower odds of AMD in the younger ages and higher odds of AMD in the older ages. And this is also true for serum vitamin D. So we consistently see this. So this tells us it's probably not specific to lutein. It's probably something to do with sampling. One of my guesses about this is that it has to do with the dilution of vulnerable genotypes. We know that having the ApoE4 allele, as about 30% of us do, is associated with higher risk of death and higher risk of many chronic diseases like cardiovascular disease and also Alzheimer's disease. And interestingly, many studies show that it is protective for macular degeneration. Is that real? I don't think so. I mean, it doesn't make sense that a condition, a chronic condition, which is encouraged by many of the same aging processes, that these others would be protective. Also, mouse models having the ApoE4 allele enhances the deleterious effect of high-fat diets, for example. So I think as we're looking at the older age groups, we have probably a lower percentage who have the E4 allele and probably other vulnerable genotypes if it's related to death. And also, if subsequently they're also changing their diet in response to chronic disease conditions, this could possibly result in the flip-flop. So this is my guess, and we are going on to study with Corinne Engelman and Kristen Myers the genotypes for people in this sample, and we'll soon be able to see whether this is the case. We're also pooling data from larger studies to international studies so that we have a large population under age 75 because we think that it's better to study samples early before the potential for these biases accumulate. There are accumulating data that suggest that there are gene-nutrient interactions with regard to these pigments and probably many other things. For example, this year published in the Rotterdam Eye study, the higher risk for having the high-risk allele for complement factor H was only observed in people who had low intake of lutein and zeaxanthin. So we'll be able to see if we observe this and other gene-nutrient interactions in the sample soon. I also want to broaden the focus right now to look at healthy diet and how that is related to macular degeneration, the extent to which these associations with lutein might reflect that. Published just this year were the fact that when you score people on the 2005 HEI, that's adherence to the 2005 Healthy Eating Index, dietary recommendations, those in the highest quintile of score have about a twofold lower odds for early AMD. And even lower odds if you score people's diets based on the Mediterranean diet scores, which are more plant-based. And dietary lutein is highly correlated with both of these scores, especially the more plant-based score. So I think, again, we really have to interpret the results in context of the overall diet patterns. It's not necessarily that these pigments specifically are responsible for the protective effect. And in fact, they may work in concert with many other aspects of diet. So since we don't know a lot of what influences the ability to accumulate lutein and zeaxanthin, we have also used this technique shown here to measure in vivo levels of the density in the eye. It's very simple because these pigments absorb blue light. This machine presents an alternating blue light, which the pigment absorbs, and green light, which it doesn't. So the respondent sees a flicker if there are equal intensities, and it's just simply changing the knob until the intensity of the two lights is the same for that person, which then can be used to calculate the density of macular pigments in the eye. And another reason for the inconsistency in studies is that varying levels of this carotenoid probably get into the eye. And of course, we can't make these plant pigments. We need to eat them. And of course, as you increase levels of these pigments in the diet, you do increase macular pigment density. But look at the scatter. We're not able to explain a lot of the density in the eye in relation to only dietary intake. We can only explain 3% of the variation by diet. And when you see a scatter like that, you wonder, well, have I measured it poorly? And so we wonder that too. And we don't think that that's the result of the scatter because there is very high retest correlation for this particular measurement. But we have found a lot of other things that might be responsible for variable ability to accumulate these plant pigments in our eye. This converges with a whole body of evidence showing that people given lutein and zeaxanthin supplements for various times don't respond with increases in the macula between 25 and 50%. And we have gone on. I'm not going to get into this in detail, but I'll simply tell you the upshot. We found that it's not only dietary lutein and zeaxanthin that predicts the levels in our eye, but other aspects of diet. Lipids. People with low fat intake do not have as high levels of macular pigment. We need lipid to absorb some fat-soluble pigments, and this is one. In fact, there have been experiments where people have eaten salads with fat-free dressing and dressing with medium amount of fat and high amount of fat. And people who have the highest level of fat on their salad dressing have absorbed more of many carotenoids, like lutein. Also, eggs have a small amount of lutein, but eating eggs can enhance macular pigment quite readily. It quite readily increases carotenoids in the blood and in the macula as well. As much as only as few as 5 eggs a day increase macular pigment. So lipids matter. It might be why we did not find more macular pigment in people in the Women's Health Initiative who were on the low-fat diet regime. Other aspects of diet matter too. It's not just all about lutein and zeaxanthin. We found that as fiber increased, macular pigment density increased. Now, what's fiber a marker for? Fruits, vegetables, grains, and those have a wide variety of antioxidants and anti-inflammatories, and so that could spare the turnover of these carotenoids. We don't know the reason for it, but that's a possibility. We've seen this in 2 samples now. Also, we and others have found in 2 samples that having lower levels of body fatness and not having diabetes are associated with higher macular pigment. We find at all levels of dietary lutein intake, people who are lean have more macular pigment than those who are obese. And similarly, we find that at all levels of dietary lutein, people who have diabetes have lower levels of macular pigment. So we've done some thinking about why is this? And there are a couple of different reasons. One is these results seem to be limited to women. We've studied women. We don't know if we'd see it in men, but 2 other groups have not found it in men. And it could be related to female ability to and propensity to accumulate these carotenoids in abdominal fat, which might be a source of these pigments for early infants. We do know that in the first few months of life, lutein and zeaxanthin accumulate are the most predominant carotenoids in colostrum. Or it could reflect that people with body fat and diabetes have more inflammation and oxidative stress. We know that to be true. That could contribute to turnover of lutein and zeaxanthin, reducing the amount that gets to the eye. Or these could be simply markers for genetic factors. And that's been a fascinating area of inquiry that we've been thinking about. So of all the things available to us in the Women's Health Initiative, all the health and lifestyle variables, we could only explain 12% variability in macular pigment by all these things. There have been twin studies since that have shown that likely genetics explain a lot of our variability in ability to accumulate macular pigment. And we have some hunches about what these might be, and even some studies that have shown some specific data along these lines in the last year. So here's our hunches. One is we might have variants which alter the degree to which we can bind these carotenoids in the eye. This is now the protein, GST-Pi1 is the protein that binds zeaxanthin. Star D3, just found this year, is the protein that binds lutein. So we suspect that variation of these might predict ability to accumulate macular pigment. We suspect variability in genes that influence transport on HDLs or metabolic syndrome, as well as variation in genes related to cholesterol uptake in the gut or retina. There's a large body of evidence showing that the export of cholesterol and the import of lutein into the eye and also into the gut is related, and then a newer body of evidence suggesting possible involvement in carotenoid cleavage enzymes. So we are waiting on the edge of our seat. Our DNA are being analyzed, and we'll be able to understand more about this in just a couple of months. But you can circumvent needing to understand these if you can get directly at macular pigment, as these people have done here. They have looked biochemically at, in autopsy specimens of eyes of people who've had macular degeneration and not, and they found that people who had macular degeneration had lower levels of lutein and zeaxanthin in their macula than those who did not. Some people say, well, this could be simply a result of having the disease process, but because they also found the same results in more peripheral areas of the retina where this disease is not affected, they argue that this argues for a causative role. Of course, it would be good if we can look at a wide variety of people, since we know how to measure macular pigment, and see how that relates to risk for macular degeneration. And to date, the data are inconsistent. It's been done in relatively small studies. This is our study here, and results have been mixed. And we've understood a little bit more about possible reasons for mixed results by this work here that was done by Tara LaRoe when she was getting her doctorate with us. Here she's indicated the odds for early macular degeneration by level of macular pigment density. Here she's shown it for quintiles 2 through 5 compared to 1. Again, she saw an odds ratio in the protective direction, not significant. But again, she sees the age interaction and sees a marginally protective association in the youngest people at risk for the condition, and no association for people that are older. So we really feel like we need to go on to understand these prospective relationships or look at younger samples. So we'll continue to do some of that. We're applying for funds to be able to look 10 years later in these people to see if macular pigment predicted lower macular degeneration. So in summary, the body of evidence suggests that diets which are high in these plant pigments, lutein and zeaxanthin, are associated with lower incidence or prevalence of nuclear cataract and macular degeneration. There's strong biologic plausibility, especially for macular degeneration. It's sometimes strong, but still variable from study to study. And as we've been discussing, not all attributable to dietary lutein. It's been observed in prospective studies, so temporality has been shown. And generally consistent across studies except for early AMD. Also consistent with rich food sources, which I haven't shown. I think an advance would be to look at people who have similarity in overall healthy or unhealthy diet scores as we try to get an idea for not necessarily just this one nutrient, but foods. Do people with very healthy diets, if they change the amount of kale they eat, have a lower risk for particular diseases, or people with unhealthy diets? So you can only do that with really big pooled studies, which we hope to do. So our future directions are these: to study interactions and joint effects, which I've alluded to. We've seen some with regard to lutein, with regard to vitamin D and sunlight, which I won't have time to tell you about. Also, I think it's important, not just in our studies, but in all studies, that instead of isolating causal elements, we start to look at them all together. Many of the things we study, physical activity, intake of a variety of nutrients, smoking, influence, oxidative stress, inflammation, hypertension. And so I think it makes sense to be putting them together than at least in addition, and not necessarily isolating the factors. The only advantage in isolating the factors is it allows us to to make a pill and have the hope of preventing a disease with one pill. And I think we've learned from beta-carotene, from folate, hopefully, that this is probably not the most important public health perspective, and it's certainly not a sustainable option. We are going to be going in other directions that investigate relation between lutein and improved vision performance, brain health, and the importance of lutein in early life. I'm going to tell you just briefly that in addition to healthy diets, overall healthy lifestyle scores are associated with even more profound lowering of odds for macular degeneration in our sample. And we'll be studying this with regard to people that are prone and not prone to AMD. So again, this supports looking at everything together, which might have a much larger impact than we're imagining. And specifically with regard to lutein, I wanted to show you that even having healthier lifestyles might help us get more macular pigment into our eyes. Here you can see that the macular pigment density in the highest quintile for dietary lutein is higher than the people in the lowest quintile by about 0.060 D units, but people with healthy lifestyle scores of 6 compared to those low levels only double the amount of lutein, but much higher level of macular pigment. So we think it all probably works together. I will stop at this place here because I want to point out that we think that lutein helps us see better, and Randy Hammond will be here on October 7th to talk about his research in this area. Hunters have known this for a while. Blue blockers, if you go to Walgreens, you put on blue blockers, you'll see that you can see contrast, but understanding the practical difference is an area of research that we will extend into. And I'm going to skip these other areas and simply say thank you for your attention. And thanks to the taxpayers who funded this through NIH grants, to the people, and to the many colleagues who contributed to this work. Most recently, Kristen Myers, who is now running the day-to-day research group. Kit Allen, who helped set up the carotenoid study when the Women's Health Initiative was in its early stages, and Corinne, who's collaborating with us with some vitamin D work. So thanks for your attention. Time for a few questions.